Hi there, welcome to the Cardiorespiratory Physiology Simulation. Here you will learn about seal adaptations that allow them to dive very deep, for long periods of time. Click Continue to start with your mission. This is our research station, here in Antarctica. Not too bad, huh? When you are ready, click on the research hut. This is our newly built research hut. My colleagues and I have recently started a research project to study deep diving physiology of Weddell seals. So far, I am loving it, and today you are very lucky. We have just captured a Weddell seal and we are going to monitor some dives. Welcome to the team. Click on the preparation area to get closer. Weddell seals can dive up to 600 meters deep and for over an hour without a breath. That is quite impressive. My human colleagues are very interested in understanding more about the differences between human and seal physiology, as they love to free dive but can only do it for a few short minutes. How long an animal can dive depends partly on their ability to sustain aerobic metabolism, which means that they are producing ATP by using oxygen to catabolize fuels in the mitochondria. During diving, without access to air, animals must depend on their oxygen stores. Let's start by taking a look at these oxygen stores. The table on the screen shows some data I have collected, and that allows to calculate normal oxygen stores for a human and a Weddell seal in the lungs. We need to estimate the total oxygen in stores inside a human or Weddell seal before it dives. So, let's calculate. Select the correct operators in the blue boxes, by clicking on them, to estimate the total oxygen stores inside a human or Weddell seal's lungs before diving. Well done. Now that you know the equation, let's estimate the oxygen stores in the various storing organs and then the total oxygen stores for both a Weddell seal and a human. We'll start with the lungs, and for that we need to multiply the volume of oxygen in the lungs by the volume of the lungs. For these calculations, the drop-down menus contain data from the table on the screen. When you select all the correct values, they turn green and the result will be calculated. Calculate oxygen in the lungs. Great. Now how about in the blood? The equation is similar to the previous one, where we need to multiply the amount of oxygen in hemoglobin by the amount of hemoglobin in blood and the total mass of blood. Click on the lab pad for more information. Super. Finally, how much oxygen is there in the muscle? Blood oxygen is stored in hemoglobin and muscle oxygen is stored in myoglobin. Applying the same principle as before, we can find out how much oxygen there is in muscle by multiplying the amount of oxygen in myoglobin by the amount of myoglobin in the muscle and the total mass of muscle. Calculate oxygen in muscle. Click on the lab pad for more information. Brilliant. Now that you know the amount of oxygen in each of the three compartments, the total oxygen stores before diving is just the sum of those. Click Calculate to add the three storage values together. So, if we now divide the total stores by the weight, we get the total stores per kilogram of body mass before the dive. This is a much better way to compare humans and seals. Calculate total oxygen in stores per kilo. Wow. That is quite a big difference between Weddell seals and humans. Seals store about 3 times as much oxygen per kilo of body mass than humans. And this is not the only adaptation these fascinating creatures have to be able to sustain deep, long dives, as we will see during this simulation. Congratulations, you just got your first piece of a puzzle, that you can see on the screen on the right. If you survive all the calculations and manage to gather 6 puzzle pieces, I have a reward for you. What is the main difference between humans and seals regarding oxygen stores? Indeed, when humans dive, we fill our lungs with air and then hold our breath. Seals, in contrast, empty their lungs before submerging. They have fewer and larger red blood cells than terrestrial mammals, with higher concentrations of oxygen-storing hemoglobin. So, they store more oxygen in the blood and less in the lungs, compared to humans. Seals exhale before diving to reduce their likelihood of experiencing bends during diving, which occurs when nitrogen bubbles form when animals depressurize. The pressure on the seal doubles for every 10 meters in depth. The other factor that determines how long aerobic metabolism works during diving is the rate at which the animal uses oxygen during the dive. Based on what we know from studies about diving in humans, let's estimate how long oxygen stores can last during a breath-hold dive. How long do you think a seal could dive for, with the oxygen in stores that you just calculated? Let's make an estimation. At this point, we don't know the actual value for the factorial increase for our seal, so we'll just leave it as a question mark on the table. Let's estimate the oxygen consumption rate of a seal during diving if it shows the same factorial increase as humans. For that, we multiply the factorial increase by the resting oxygen consumption. Later on, after some measurements, we will come back to this and see how close to reality our estimation was. Choose the correct values. We now know how much oxygen there is available in each store, as well as in total, and we have just calculated a hypothetical oxygen consumption for the seal if it shows the same factorial increase as humans. So, what is the equation to calculate the aerobic dive limit in minutes? Yes, the time it will take to exhaust the total oxygen in stores can be obtained by dividing the amount of oxygen by the rate of its consumption. Choose the correct values. So, you now have an estimate for how long seals can dive aerobically if they show the same factorial increase in oxygen consumption as humans during diving. About 10 minutes, compared to roughly 1 minute and 24 seconds for humans. Another factor that can affect the duration of dives is the animal's capacity to generate ATP anaerobically, and to tolerate the acidosis that occurs when lactate is produced. So, we will also measure lactate levels in the blood to see if there is any indication that seals are using anaerobic metabolism when they dive. Let's have a look at the seal, that is right here, and do some experiments. My colleagues have captured it using nets and they are taking good care of it, checking that it is breathing and moistured. You are just on time to watch and help out. All necessary instruments and equipment are on this workbench and by the hole. The seal has been previously anesthetized and weighed. Click on the camera to pick it up. Click on the seal to put the camera on it. Now click on the instruments pack to pick it up. Click on the seal to place the instruments on it. The video camera can be used to measure foraging and the instruments package includes an electrocardiogram system. Other things could also be measured, such as swim speed with a flow meter and flipper strokes with an accelerometer. Click on the catheter to put it on the seal. Excellent. This will make taking blood samples quick and easy. Now we need to put the seal in the water. We will need to take a blood sample before diving, so that we can compare the lactate levels now and for the dives we are monitoring. Get an empty blood collection tube from the rack on the workbench, click on the adapter to mount it and then on the seal to collect the blood. Collect a blood sample from the seal. Click on the dome to cover the diving hole. Air is now flushing through the dome at a known speed. I have just calibrated and zeroed the flow meter and oxygen analyzer for you. Air from the seal goes through a column that removes water vapor and carbon dioxide. Then, it goes through the flow meter and oxygen analyzer and finally outside. In just a second, we will look at the plot of oxygen fraction versus time on the wall screen. At the same time, gas from the cylinder is being passed through the other column, and the pump is sending it into the dome. With these two systems, in and out, we can measure the amount of oxygen flowing into and out of the dome. Before the dive, for the resting seal, with a flow rate of 200,000 milliliters per minute, the oxygen fraction is now starting to stabilize. Have a look at the data table on your lab pad and a graph will appear on the screen soon. Let's calculate the resting oxygen consumption rate. For that, we need to know the values of oxygen entering the dome, Fi, and leaving the dome, Fe. The Fe value should be taken from the flat part of the graph. What is the oxygen level with no seal under the dome? Precisely, the seal is already in the hole and looks like it might dive at any time, so let's calculate the oxygen consumption rate before diving. Click on the wall screen. Well done, you now have the initial value for the resting oxygen consumption rate. Let's have a look at an ECG, received from the instruments pack. On the screen, you can see the heart rate that was monitored for 10 seconds. What is the heart rate of a resting seal? The image on the screen corresponds to 10 seconds. Exactly, 11 beats in 10 seconds correspond to 66 beats per minute. The seal has just gone in. The instruments will be collecting data while the seal is gone, and soon you will be able to see the values on the screen and on your lab pad. It has been 12 minutes. Let's see the data. The table is on your lab pad and the graph is on the wall screen. Note how the curve shape has changed from almost flat to this U shape. Choose the correct values. Now we need to collect a blood sample for the 12-minute dive to monitor the lactate levels. Click on the dome to access the seal. Collect a blood sample from the seal. Click on the dome to cover the diving hole. The seal has just gone in again. It has been 30 minutes. Let's calculate the oxygen consumption during these 30 minutes. Again, note the shape of the curve and how it is changing with the duration of the dives. Choose the correct values. Wow! In a 30-minute dive, the seal has consumed oxygen at about the same rate or even a little bit less than during a 12-minute dive and even at rest. That is impressive. Humans would have consumed more. Factorial increase must be very different for these two species. We need a last blood sample for the 30-minute dive to monitor the lactate levels. Click on the dome to access the seal. Collect a blood sample from the seal. Click on the dome to cover the diving hole. Do seals show approximately the same factorial increase in oxygen consumption as humans during diving? Indeed, and this allows them to dive for much longer than humans. With a 30-minute dive, did the rate of oxygen consumption during diving change relative to the 12-minute dive? Exactly. We have just seen that seals have a much lower factorial increase in oxygen consumption during diving compared to humans. All data are already available for analysis in the system. You can now remove the instruments and the seal will be allowed to recover for a little while. Then, my colleagues will check on it and release it back into the wild. Click on the dome to access the seal. Click on the seal to remove the instruments. We have monitored the heart rate of several seals during dives and the pattern tends to be very similar each time. How does the seal's heart respond to diving? Yes, lower metabolism allows for longer diving times. What do the heart rate and oxygen consumption rate patterns suggest about seal diving? Exactly, lower heart rate contributes to less oxygen consumption which, in turn, helps saving energy. I have just analyzed the blood samples and there was no lactate accumulation for the 12-minute dive. For the 30-minute dive, however, lactate rose from initial 2 mmol divided biliter at rest, to 10 mmol divided biliter. It looks like ATP used for diving was being generated by two different mechanisms during these dives. The 12-minute dive seems to have been completely aerobic, as there was no lactate accumulation, and the 30-minute dive seems to have been partially anaerobic. To turn these thoughts into numbers, let's calculate ATPs generated for the aerobic dive, from oxygen, and for the partially anaerobic dive from lactate. Let's start with converting oxygen to standard units of temperature and pressure. Click on the screen to see the values from the data we just collected and finally click Calculate to obtain the result. Convert oxygen to standard units. Well done, the units are now correct to proceed with the calculations. So, we can calculate how many molecules of oxygen were used during the dive. Calculate molecules of oxygen used. Finally, we can calculate the ATPs generated from the aerobic metabolism by multiplying the number of molecules of oxygen by the number of ATPs produced per molecule of oxygen consumed. There you have it. Moving on to the anaerobic metabolism, let's calculate how many millimole of lactate were produced. Calculate the lactate produced. Great. So, we can determine how many molecules of lactate the seal has accumulated, by multiplying the lactate produced by Avogadro's number. Calculate molecules of lactate. And with that, finally, how many ATPs were generated during anaerobic metabolism. Calculate ATPs generated by aerobic metabolism. This is great. Now you know the ATPs generated by both aerobic and anaerobic metabolism. You are awesome and you deserve one more puzzle piece. What fraction of all ATP production is produced by anaerobic metabolism during a 30-minute dive? Look at the ATP values on the screen, round them up and try to estimate approximately what percentage of the total was generated by anaerobic metabolism. That's right. During this partially anaerobic dive, about 20% of ATP was generated by anaerobic metabolism from lactate and 80% of ATPs were generated by aerobic metabolism from oxygen. The 12-minute dive was completely aerobic, as there was no lactate accumulation. Now that you've learned so much about the physiology of diving, what do you note as major adaptations of seals to enhance their ability to dive? Select the incorrect adaptation that seals have to improve their oxygen storage capacity. Indeed, seals have much larger lungs than humans because they are bigger animals, but as a percentage as their total body mass, seals have smaller lungs than humans. Now that we've collected all the data, do you remember that assuming the same factorial increase for humans and seals, we estimated that the seal could dive for 10 minutes using its oxygen stores, while a human would only be able to dive for 1.4 minutes? Let's go back to that equation, and recalculate the aerobic dive limit for the seal. Wow, that is quite different from your initial calculation, more than double the time. No wonder my human colleagues are so impressed by seals' ability to dive for so long and also very jealous. This seal can dive for over 16 times longer than an average human can. You are doing great. Here is your fifth piece of the puzzle. Is your calculated aerobic dive limit consistent with the observed pattern of blood lactate? You got it. Seals can dive for much longer than humans due to their specialized physiology, which allows them to perform both aerobic, as well as partially anaerobic dives, when the aerobic dive limit is exceeded, as we just observed for the 30-minute dive. Awesome, you are now an expert on cardiorespiratory adaptations of seals to deep diving. With this answer, you get your sixth and final piece of the puzzle. With it, you have earned a special dive with a seal. I hope you enjoy it. Click on the seal. Seals can dive very deep because they have special adaptations. Oh, look, there is one coming our way. Unlike myself, seals glide to save energy. I'm more of a freestyler. Click on the seal's lungs. The lungs are reinforced by muscle and divided Buerr cartilage and able to compress under pressure, which avoids bends. This is quite a handy adaptation to have when you are diving as deep as a seal does. Click on the seal's heart to learn more. The heart rate drops to reduce oxygen consumption and preserve energy. Click on the seal's blood vessels, near the heart. Seals have a big volume of blood and most of the oxygen is stored there, in hemoglobin. They also have a high hematocrit compared to humans. Wow, swimming with seals is so much fun. Thank you for your help today collecting data and making all those calculations. It was great learning about cardiorespiratory adaptations and physiological responses to deep diving. Now I can tell my human colleagues all about the amazing physiological adaptation seals have that explain why they don't need scuba diving gear to dive for up to 30 minutes, even when they've gone beyond their aerobic dive limit. Your score has been saved together with your 100% progress. Congratulations, you have completed the simulation. We hope you enjoyed it and you feel wiser now.